So today what we're going to do is do a very quick review of the auditory system. We're going to play the auditory game, and then we'll turn our attention to the olfactory and the gustatory systems, our ability to detect chemicals, either by smelling them or by tasting them. What I'm trying to do is pull up. Game. Okay, so here's the game. Who wants to start? Good. Far corner, we'll just work our way across and then zigzag across the room. Okay, so you know how to play the game. You point to a structure, point to a button, and then tell me what word I should put there. Cochlea.
That's the tympanic membrane.
Yes, it is. Oh. What did I get? Tympanic cavity.
I missed this lecture, so rest of your low.
Yeah, good.
The round window, I think is maybe that one down there. I can't see well.
Yeah, it's hard to tell. Round window.
Or is it up?
Is this the one you wanted me to try?
Yeah, try that one. Or the one above it?
The really top one? Yeah.
Oh, is it the really top one?
No, that one?
I think so. So let's choose a different one. Let's
Then the stapes, I can't see that right now.
Stapes?
The stapes, it's the stirrup. So I don't.
Which button?
I'm not sure which one it points to.
Okay, yeah, it's really hard to see. If I dim the lights, will that help?
Yeah, probably.
Okay, let me try that.
I think it is the top one.
Then try the incus. I think
Okay,
it's.
incus.
I think
Which one?
Try that one.
Okay, good. So, it's not easy, and I mean, your visual acuity makes it difficult.
I'm guessing auditory too is represented.
Auditory 2?
Yeah.
Yeah, we didn't talk about that, so don't pick that one.
Okay, I'm correcting that.
The oracle? You want this one? You want the auditory canal? I can't hear you.
Yeah.
Okay, what button? Can't go that far.
Probably, I'm not sure, here.
Okay, auditory canal? I don't think so.
Oh.
That's a muscle. I'll give you that one. That's the tensor tympani
That's
muscle. We
it.
didn't talk about that one.
How about here?
Auditory canal.
I'm not
This
sure.
one here? I don't think so. Nope, that's the auditory tube.
Yeah.
Okay? One more chance. I mean, and you don't have to do auditory canal. You can pick something else.
Try the oracle.
Okay, oracle? I don't think so, no. The oracle is the name for the outer ear, so my guess is it's this one. Could be this one. No, that's the helix, so this is the oracle.
I don't know.
The oracle is the outer ear.
Okay.
Malleus? Which one? Malleus, good. Semicircular ducts, right.
Is one of these the vestibular?
Vestibular nerve, yes, one of those is. Which one do you think it would be? Okay, you got a 50-50 chance. Yes, that's right. The vestibular nerve carrying information from the vestibular apparatus, I won't give you the names except for the ones that you've already gotten, the semicircular canals and the other two structures of the vestibular apparatus. Who's got the laser?
So this is the cochlear nerve.
Cochlear nerve, right. The cochlear branch of the vestibulocochlear nerve, really, carrying information from the cochlea.
Is that the same?
That's what?
The same.
Yes, that's the stapes.
I know we said something before, but I'm sure that would be good, or I think that would be good. It's wrong there. That was
This one here? You're right. Okay. I didn't see what it was pointing to. Okay, so
I
you were
figured
right.
it was like
Whoever got it the first time around, you get thumbs up anyway. Not with me.
The auditory canal is this one.
Yep, the auditory canal is the tube. It's not a tube, it's the canal from the external ear to the tympanic membrane. Tympanic cavity, which one? Yeah, that'd be that one, right? And the helix is the name of the top of the auricle. Okay, let's do some more. Which more? You don't have to feel left out.
The cochlea.
Okay, cochlea, I mean, there's some cheap ones. Which one did you want?
The bottom.
Yeah, okay. That's good, that's right. Cochlear nerve is where? Yeah, that's right.
I don't know how to pronounce this one.
We only covered some of these, so for example, I didn't ask you to know which of the semicircular ducts is the lateral one. I think it's this one. So I'm gonna clean this up a little bit. The anterior, I don't know, maybe this one. And the posterior. I am not asking you to know which one is which. You know, the name semicircular ducts or semicircular canals is sufficient. You don't need to know anterior, posterior, or lateral. Vestibular nerve. Which one? Yeah, let's try that one. Okay. Vestibular branch of the vestibulocochlear nerve. Wait, you already went.
Yeah,
I mean,
we
well,
can
it's
go
true, you
on.
got cheap one, you got the earlobe, you can go again.
Okay. Is this the vestibular ganglia?
Okay, vestibular ganglia, this one here? Is that the one you were choosing?
The one below it.
The one below it. Good. I didn't teach that one, but you figured it out. Vestibular nerve has a vestibular ganglion.
Is this one the
Saccule, right. So the this one is, which
Yeah,
one did you?
yeah, that
Okay.
one.
Yes, the saccule is the one that's closer to the cochlea. What is that? This one?
No.
Oh. What name?
The other sac.
The utricle is the other the other one. And that goes here. Is that what, I'm sorry. I'll give it to you. That one goes there. Okay? Utricle and saccule are the parts of the vestibular system that detect lateral acceleration. Circular, semicircular canals detect rotational acceleration. Is there anything else here that we need to know? Ampulla. We did cover ampulla. Where's an ampulla? Somebody Who's got the laser?
Me.
Aiden, it's yours. Point, let's just choose, which one is ampulla?
I was going to do the spinal ganglia, which actually would be
Okay, choose spiral ganglion.
that one, right?
Yes, okay?
So,
Anybody wanna, who hasn't gone yet? Okay, Leah,
thank
you
you.
wanna choose ampulla?
I'm guessing you feel like it's like the top left.
This one? Nope.
No.
So the ampulla, remember the ampulla are those bulges in the semicircular canals that hold the cupula, and so the other one is the endolymphic sac. So we don't have to do this anymore, that's enough. We can go into the organ of Corti. There is actually some games to play on the anatomy of the eyeball here and here. So there's a bunch of these that are listed in your PowerPoint. You can play the game. Okay. It's a good review, but remember the coloring book and the key concepts list is your assignment. Okay, so let's get on with chemical senses. That's where we're going today. We can detect chemicals. We detect them in our nose through olfaction, and we detect them in our mouth through the sense of taste. Let's talk about how we do that. And we'll play another game. I'll talk while this is going around. Here's a set of odorants, and the name of the odorant is covered up. So what I want you to do is, but there's a number on it. As we're talking, smell the odorant. Please do not spill it, because they will hate me in this building forever if you do. So don't spill it. But smell the odorant and write down what you think it is, the name of that odor associated with the number. And then after everybody gets a chance to play the game, I'll start number 3 going there in the reverse direction. And so smell the odor, don't talk about it, just write down what you think it is, and if it triggers any specific memories. Does it, rather than just what it is, there's 2 things. Does it remind you of something? Okay? So go ahead and pass those around, smell them, write down your ID, and write down if it reminds you of anything. And then at the end of the class, we'll talk about it. So we can detect chemicals. We have ability to transduce odors and tastes into electrical information that is transmitted by action potentials and action potential frequency to our brain that our brain interprets as the smell of whatever it is. So you know that the sense of smell is transduced inside your nose. There is an olfactory epithelium at the top, at the roof of your nasal cavity. That olfactory epithelium is right next to, but separated by bone from, the olfactory bulb. The olfactory bulb is on the superior surface of a bone. You saw it in lab on the inside, it's part of the brain, but the olfactory epithelium is inside your nose. So there's a bone, a special bone called the cribriform plate that separates. So here's a cartoon of the cilia of sensory neurons that send their axons to the olfactory bulb. Okay? Just use I use some keywords. Cilia that contain the receptors for odorants of sensory neurons with cell bodies that send their axons to the olfactory bulb. Are these long or short receptors?
Long.
They are long receptors because they have axons. The axons are not very long, receptors. Not as long as a sensory neuron in our fingertip, but they are still considered long receptors because they have axons.
So is that the only difference then, when they have axons in them?
That is the difference. That's the key difference. And this yellow structure here in this cartoon is supposed to illustrate a perforated bone at the base of the skull called the cribriform plate.
So, you said the nerves go through that bone?
Yeah, nerves go through that bone. And then make their first synapse in the olfactory bulb. The first synapse of the olfactory system is in the olfactory bulb. So an odorant enters your nose, binds to receptors on the cilia of olfactory sensory neurons, and that's going to cause the electrical change that we'll talk about momentarily. Here's another cartoon of it. Here's the olfactory epithelium in the nasal cavity. It's got mucus that we're not going to talk too much about. There are these fine cilia of the olfactory neurons that branch out into the mucus to give it lots of surface area to detect those odorants. The cell bodies going through bone. I'm sorry. So these were supportive cells. Here's the bone, the cribriform plate. The axons go through the bone and form their first synapse in the olfactory bulb. The olfactory bulb is organized into what are called glomeruli of specific synapses, and we'll talk about that in a minute. Then the secondary neurons. So these are the secondary neurons that have been activated by the primary neurons. The secondary neurons send their axons to the brain. Well, strictly speaking, the olfactory bulb is part of the brain, but it goes into the cortex. This is just an example of 7 or 8 different chemical structures of things that we can smell. So, an almond smell, that's a structure that smells like almond. And here is a structure that smells like skunk. It's got a thiol. Thiols have a skunky smell. And here's a rosy smell, and here's a gingerine smell. I'm not going to ask you to try to remember which chemical triggers which smell. Of course not, because there's 10,000 of them, and it would be pointless. And in fact, it's impossible to try to predict what something will smell like based on its structure. Here are 2 structures that are almost identical. They are enantiomers of each other, and yet they smell very different. The smell of spearmint and the smell of rye bread, caraway. From almost the same structure, from enantiomers of the same structure. Kendra?
I was just asking what is caraway?
Caraway is the seed that gives rye bread its distinctive odor. So how do the olfactory cilia, the cilia of olfactory sensory neurons, transduce the binding of an odorant to an electrical signal that generates action potentials in its axon? Here is that story. Here's the receptor. It's got 7 transmembrane spanning regions. It's a GPCR. An odorant binds. What happens? Conformational change. Okay? What happens then? Conformational change of the alpha subunit of the G protein. This G protein has a name. Just remember, the G protein in the retina is called transducin. The G protein in the olfactory epithelial cell, the olfactory sensory cell, the cilia, has a name, and it's called G-olf. It's not golf, it's G-olf. You can remember it as golf, but don't tell me it's golf, tell me it's G-olf, okay? It's the olfactory G protein. It's related to one of the G proteins that has letters, S, I, or Q, and you'll see which one in a moment. Okay, GDP dissociates, GTP associates, GTP bound alpha separates from beta gamma. This should sound familiar. I don't know how many times we've done this. the activated GTP bound alpha binds to adenylyl cyclase. Which G protein is G-ULF most similar to? GS. It acts like GS. It is not GS, but it acts like GS. What does adenylyl cyclase do when it is activated by GTP-bound G-ULF? Produces cyclic AMP from ATP. What does cyclic AMP do here? Does not activate pKa. Now the story deviates. The second messenger activates a cyclic nucleotide gated cation channel. Cyclic AMP activates a cyclic AMP gated or a cyclic nucleotide gated ion channel that is permeable to cations. So what does the opening of this channel do to the resting membrane potential? It depolarizes it. It's no longer at rest. It's been activated by the odorant, so maybe that threw you off. Opening of this channel, opening of these channels causes influx of cation and depolarizes it. This information could be testable on exam 2, just as the way it was on exam 1. So don't jettison it just because we moved on mostly. We're back to the same old, same old. At opening of a cation channel, cation influx, depolarization. Depolarization past threshold is going to increase the triggering of action potentials and increase action potential frequency in the axons of this cell. We're not seeing this. We're not seeing the axon of the cell because this is a highly magnified cartoon of the cilia. But the cilia, depolarization of the cilia, depolarization of the cell body, depolarization of the spike initiation zone, activation of action potentials that propagate to the olfactory bulb. So there it is. There is the mechanism of transducing an odorant, something that smells, you are experiencing it right now, something that smells, into electrical activity that goes to your brain and triggers memories or triggers names using declarative memory processes that we're not going to talk about in this course very much. Kendra?
So is smell processed by the amount of the sound that open?
Oh, it's way more complicated than that. So smell is detected by the binding of the odorant to a GPCR. That triggers a cascade of events that causes opening of channels, that causes a depolarization that leads to an action potential frequency. Now, information is going to the brain. So now let's unpack the next step a little bit, but it's way more complicated than what you just said. So, before I do that, let me just talk about these odorant receptors. We have something like 350 different odorant receptors. Our genome codes for 350 different odorant receptors. A dog has more, a rat has even more. We have something like 12 million odorant receptor cells. So 12 million cells are expressing these 350 different odorants. Each receptor cell expresses one type of odorant receptor.
So you said that, and then this triggers that G protein pathway you're looking at.
In that cell.
So is each receptor, you're saying each physically individual cell only codes for one specific?
Each individual receptor cell expresses, it's got the DNA for them all, but it only expresses one type of odorant receptor.
So theoretically, these should be able to replicate if some die.
Yes, our olfactory epithelium does usually regenerate. There are conditions called anosmia where the odorant receptors die off and they don't regenerate, and those people can't smell anything.
How long does it usually last?
Couple days. So we have 350 different odorant receptors. They're GPCRs, so they have 1, 2, 3, 4, 5, 6, 7 different transmembrane domains. They're all almost exactly the same, but there are some, so the blue residues here, the blue amino acids, are highly conserved. The red amino acids are highly variable. And it's the variable amino acids that give each odorant receptor its special characteristics of interacting with a specific odorant. This is sort of like the story of the color receptors in the retina, right? The color receptors in the retina are all GPCRs, but they all have a few different amino acids that tune them for different frequencies of light, different colors. Here we have quite a few different amino acid variants, the red ones, that give each receptor its ability to interact with a specific set of odorant molecules. Now you're probably wondering, wait a second, we've got 350 different odorant molecules, and yet we can detect something like 10,000 different odorants. We can discriminate 10,000 different odorants with 350 different receptors. How is that possible? And it's possible because each odorant receptor responds to a spectrum of odorants. And then each odorant triggers varying activity of a set of receptors. So this is a combinatorial function, right? So if you smell, for example, octanol, where's octanol? C8OH, there's octanol, C8OH, that is the structure of octanol. It smells orange rosy. It activates 4 different receptors, one of them stronger than the other 3. That combination is what we interpret as orange rosy. Octanal, which one's octanal? I'm not sure octanal is on here. Octanoic acid, carboxyl acid, C8COOH. Actually, this is octanoic acid, right? 8. It activates a slightly different, let's just look at C8OH and C8COH. It activates an overlapping but slightly different pattern of receptors. And so we can discriminate between a sour, fruity, rancidy smell versus a fresh, citrusy, orangey smell by using these different combinations of receptor activations. So we can turn those 350 different receptors into the ability to discriminate between tens of thousands of odorants because of the combinations. Yes?
Do we have like more receptors for like certain kinds of smells, like particular bad smells, or ones that could be dangerous to us? I feel involved in that way or no?
That's a good question, and I don't know the answer to that. Whether we have more receptors for potentially dangerous things like skunky smells than, nice, fruity, flowery smells. I don't know the answer to that. You could imagine that we would have evolved a better ability to discriminate between things that are dangerous, things that represent spoiled food, sourness, and things that represent tasty food like fruit. But I don't know the answer to your question. So now, let's talk about our ability to detect smell in our brains. There is a part of the brain called the olfactory cortex. So those axons, the secondary axons from the olfactory bulb, right? The first axon is in the the first synapse is in the olfactory bulb. So the secondary cells send their axons to the olfactory cortex, which is in the medial parietal lobe. So this looks like it's part of the temporal
Well, actually, it's on the border between the temporal and the parietal lobe. It's very close to the hippocampus, which is interesting because odor, you may have heard of the book Remembrance of Things Past. It's a huge French book, a book by a French author, Marcel Proust. It's this huge book of his memory of his life. But it starts with he smells a cake. He's at a coffee shop and he smells a specific type of cake. And this entire wave of memory that he writes about was triggered, in the story, is triggered by the smell of a cake and dipped into tea. And that smell triggers this wave of memory. And it's true that smell is associated with memory in a way that not all other senses are. Certainly not touch. Maybe sound. You remember a song, that triggers a memory. But smell does it too. We haven't talked about the taste cortex yet. We're talking about the olfactory cortex. So this slide is actually a pretty good review slide of the anatomy. I'm not going to go through it, but you could go through it to remind yourself of where is the somatosensory cortex? Where is the primary visual cortex? Where is the primary auditory cortex? Here in the superior temporal lobe. So this is a pretty good review. It's got some other information that we haven't covered yet, like the primary motor cortex, which we'll do after break. The primary olfactory cortex is in the medial boundary between the temporal lobe and the parietal lobe. But smell goes everywhere in the brain. So this is an actual MRI of fMRI, a functional MRI of somebody's brain. So we're looking at it sort of obliquely from the top. What's the name of this structure? That's the corpus callosum. It's got a very distinctive shape. You should be able to make it out in just about any picture in any orientation that I throw at you. What's the name of this structure? What's the name of the white matter that is underneath the corpus callosum? It begins with an F. Fornix. Okay, so I don't have to throw this at you. It's too close to break for me to penalize, to punish you with that. But there's the cerebellum. There's the cortex, frontal lobe, occipital lobe. That's probably the calcarine fissure right there. Da-da-da-da-da. But here's a slice through the temporal lobe of this person. So it's there's a vertical angle and a horizontal angle, and you can see areas of the brain that were activated by the smell of a rose. This is, of course, an artistic rendition of what was happening. This person was in an MRI machine, and they probably smelled some odorant. Probably not, or just a flower. So even though smell starts in a specific area, it quickly goes into lots of areas of the brain, and some of those are associated with memory, triggering memory. Let's take a short break, and we'll come back and talk about taste. And while we're taking the break, we can, if you should have on a piece of paper your answers to the 5 different odorants. If you don't have all 5, somebody just say, I need number 3. Who's got number 3, etc. We can do that during the break. 4, 5. Odorants. So somebody give me the names of what you wrote odorant 1 was, the name of odorant 1.
I
Matt.
put Vicks lotion.
Vicks
My mom used to use
lotion.
Okay. Spearmint gum.
Spearmint gum. Spearmint. Okay. This is number 1.
Mm-hmm.
Oh, no, never mind.
Yeah.
I know. I was like,
Number 1.
I said it's not country.
Evergreen.
Said oregano.
Oregano. Oregano, evergreen, Vicks,
I got a little bit of eucalyptus out of that.
eucalyptus, winter. Okay. So what this is it's a pine needle extract. So this is the smell of pine trees. But it's interesting, isn't it? How much we use other cues. You would never mistake the smell of eucalyptus for a mistake of a pine tree, because you can tell the difference between a eucalyptus tree and a pine tree if you're out in the forest. And so smell, we have a very fine sense of smell, but it's not acutely associated with our word, are associated with words. Somebody said forest or something, right? That was the closest one. Okay, number 2. What is your name for number 2? Aiden.
I feel like Pfeffernuss.
A what?
Associated with Pfeffernuss.
I don't even know what that is.
Me neither.
I said rose.
Rose?
A type of
I said
oh,
Lemongrass? See, okay, we're getting into some cultural differences here, I think. Yes.
Said baby powder.
Baby powder, okay.
Hey,
So this is the smell of rose water. This is a rose smell.
Oh, I think I never did that. Okay.
So, did anyone trigger did number 2 trigger memory as a, what did,
It smells like a dessert that we make back at home.
Okay.
Like they specifically put like rose extract in it.
so it reminds you of a dessert. Anybody else? Yes.
It smelled like bug candles.
Bug candles. That's interesting. It smelled like summertime bug candles. Yeah.
I mean, yeah, like Lauren said that, and
You
I
have
helped out, and my
citronella
aunt not only uses like rose water spray in the house, but also uses the bug's
Oh,
peas.
so that you've associated
So both
those 2
the
odors
harvest
together.
Or my aunt's house.
Yeah. Okay. Number 3. Anybody want to name number 3? Maddie.
Clove.
Clove.
Oh.
Hmm?
Tea with milk.
Tea with milk. Hmm?
But hold on, I said Pfeffernuss for number 3, honest.
Okay.
I called it peppercorn because I was thinking of the little black thing.
Oh, those are cloves.
Yes, I
Because this is
peppercorn.
clove oil. This is the extract of cloves. And so, if any of you have any of you ever had dental work where they use clove oil as the anesthetic?
No.
So for a set of people, myself included, the smell of clove oil is now indelibly associated with tooth pain. Because they packed my tooth with clove oil to try to calm the pain of a inflamed dental nerve that was causing intense pain. So I had to have a root canal, and then they packed it with clove oil. So clove does for me is sort of, it smells, what are some of the memory, so you don't have the pain memory of clove, what are some of the memory that you got, that cloves trigger?
Again, this is the Pfeffernuss type thing that I was.
Pfeffernuss.
So making.
Is that a cookie?
Yeah, sort of.
Okay.
People know what that is.
No.
Never heard of it.
I'll show you
A cookie.
after class. It's like.
Maddie?
My grandma used to burn clove candles in her house.
Clove
and I was
candles, so it reminded you of grandma. Lots of people, it reminds of Christmas. You know, the smell of cloves and orange for Christmas. Number 4. Anybody give me a name for number 4? Lemongrass,
Just lemon.
lemon,
Lysol.
Lysol. So this is the smell of lemon, right? This is a lemon extract. So lemon, lemongrass, lemon extract, Lysol uses. This is a fragrance that is commonly used in cleaning products, right? There's a whole industry of perfumery associated not just with perfumes that we put on our bodies, but perfumes that perfume our, cleaning products and what else? Well, especially cleaning products, but laundry detergents, right? The smell of laundry detergent, unless you buy the odor-free, fragrance-free they call it. There's an industry associated with putting in the fragrance of Tide versus Gain detergent. And number 5.
This looks like
Spearmint. Mint. Yeah, so this is peppermint. Have you ever heard of this soap called Dr. Bronner's?
Yes.
There's this liquid soap that is intensely minty. And so for some people, this triggers the memory of showers and baths. Anyway, it's interesting how sometimes difficult it is to identify the odor, even though it's easy to detect it. It's just an interesting phenomenon. Okay, we're going to transition now to taste, our other way of detecting chemicals, using our taste buds. So unlike odorants, where there's something like 10,000 different modalities, there are a limited number of taste modalities. We mix them, but And we use odor for flavor. Many of our flavors are associated with odor because, after all, our mouth is connected to our nose, and so a lot of the volatile compounds that are released from food when we're chewing go to our nose and trigger flavor responses. But the taste buds themselves, in our tongues and in the back of our throat, the taste buds themselves have a limited number of chemicals that activate them. So there's the smell, the taste of sweetness, not just glucose, of course, anything that tastes sweet, salty. Umami is the name of, umami is the Japanese word for delicious or savory. And it's actually the taste of glutamate. So when they put monosodium glutamate into food, they, hopefully not you, are trying to activate your umami receptors. We taste bitter, and we're pretty sensitive to bitter because lots of toxins are bitter. And so we're pretty good at discriminating bitter taste. Now, some bitter tastes good to us, and we've learned, like, coffee is a bitter taste. Children typically can't imagine a single reason why their parents would drink coffee, but we learn to like the taste of coffee. And we taste sourness, acid. Those are the 5 taste modalities. Some say capsaicin, the taste of spiciness, is a 6th taste modality, but it's really the pain temperature modality. So, for some cultures, obviously, this is a taste, not a pain receptor. And so, for some cultures, we would have to be open-minded about a 6th taste modality. But 5 is universally true. 6th is, this is true. It is a taste modality for some, but not all people. So we have taste buds. You all know that your tongue is studded with taste buds. You may have heard that we only taste that we taste sweetness with the tip of our tongue and we taste bitterness with the back of our tongue. And that's not true. It is true that we have more sweet taste receptor expression at the tip of the tongue, and less bitter taste receptors at the tip of the tongue, and more bitter and less sweet at the back. So the statement that we taste sweet with the front and bitter with the back comes from some truth, but it's an exaggeration of the truth. We taste sweetness better with the tip, and we taste bitter better with the back. But we taste both in both places. There are different names for the different types of taste buds that I don't want you to learn. All taste buds are little sort of openings, some with a bump on the surface and some without a bump on the surface. Openings in the surface where a tastant molecule gets in and activates receptor cells in the taste bud. Each taste bud can express a multiple number of receptors with multiple taste modality alignments. The receptor cells have receptor molecules that bind the tastant, and the receptor cell then converts the binding of that tastant into an electrical signal. And so you can imagine we're going to unpack that in about 2 minutes. The receptor cell releases neurotransmitter that activates the afferent neuron that goes to the brain, and the taste afferents are in the glossopharyngeal nerve, the vagus nerve, and the facial nerve. You may have remembered from your memorization of the cranial nerves that several of them carry taste information from the mouth. Are taste receptor cells long or short receptor cells?
Short.
They're short.
They do not have an axon. The first axon is of the secondary cell. The first synapse of the taste pathway is in the taste bud. So let's go through the modalities, or the mechanisms, of how we sense taste. Okay? I'll start with the easiest one first. What I hope is the easiest one first. Sweet, bitter, or umami activate a GPCR, activate different GPCRs. So there's a sweet GPCR, a bitter sensitive GPCR, and a umami sensitive GPCR. They're all lumped together because after that the story is the same. Tell me, come on. You tell me the story. Binding of the tastant to the GPCR. Next. Thank you. Conformational change. Then
You have the exchange of GTP with the GDP.
Conformational change, and then GDP dissociation and GTP association of the alpha.
Yeah, then the alpha, so
Alpha then, the activated alpha with GTP bound then activates phospholipase C in this story. What G protein is the taste pathway activated? GQ. This is GQ. Phospholipase C does what? Maddie? Hydrolyzes PIP2 into diacylglycerol and IP3. IP3 does what? Diffuses to the endoplasmic reticulum, activates an IP3 gated channel. Calcium is released from the endoplasmic reticulum. Okay, now, do we see PKC on this story? No. Do we see calmodulin on this story? No. Calcium activates some channels. We've got calcium activated. Let's just focus on this one. Calcium activated sodium permeable channels. What happens to the taste cell, the receptor cell? Depolarization, activation of voltage-gated calcium channels, calcium influx through voltage-gated calcium channels. So now we've got two ways that calcium increased in the cell. We've got the calcium from the IP3 receptor, and we got the calcium from the voltage-gated calcium channel. Synaptotagmin, snares, neurotransmitter release. That story is just, again, a review of the canonical GQ story. We didn't have to get into PKC, we didn't have to get into calmodulin as we have in the past. All we needed to remember is that we activated a ligand, a calcium-activated cation, sodium permeable channel.
So does the GPCR pathway, does it stop at the second messenger to call it a GQ or GS?
So which is the second messenger in this pathway? Is that your question?
No, do we call a GPCR pathway according to where the second messenger stops? So if the second messenger is not the same as, let's say, GQ pathway, we don't call it the GQ pathway?
We would still call it the GQ pathway because we're still activating GQ. It's just that in these cells, the calcium does not activate PKC or calmodulin.
But this is a GQ.
This is a GQ pathway. Kendra. Oh, so let's not worry about this thing. There's another channel that releases ATP that actually activates another receptor. It's a whole nother story that this slide has that I would rather not get into. So let's not worry about that. Yeah.
If you have calcium and you have, I used to like, dad has it, like, if you used
diacylglycerol and calcium release.
Right, like why would they not enter to activate PKC? Is it just not present in like?
It's not expressed.
Okay.
And there might be some PKC expressed that is activated by the combination of diacylglycerol and calcium. Then we could be activating some PKC, but that activation is not required for the depolarization sufficient and the calcium signal that triggers neurotransmitter release. Okay, so that's how we transduce, right? We went from the binding of a tastant to the release of neurotransmitter in the receptor cell via this pathway, a GQ coupled pathway. So we've gotten 3 out of the 5 out of the way. Salt is the next easiest one. And that's all that you need to know for how salt, sodium chloride, activates salty taste in your mouth, is that there is a channel in the taste bud, in the salt sensing taste receptors, called an epithelial sodium channel. ENaC is the abbreviation for an epithelial sodium channel. This is a channel that is constitutively active. It doesn't require a stimulus to open. But if there's lots of salt outside, there's more salt that goes through it and more depolarization. So adding sodium chloride to the outside of this cell, which has a relatively high expression of a sodium channel, allows depolarization. Depolarization, calcium influx through voltage-gated calcium channels, synaptotagmin, snares, neurotransmitter release. So that one, in some ways, that's easier than this. All you have to do is remember that there's a salt channel, sodium channel. This is not the voltage-gated sodium channel that is the same channel that triggers the action potential in the afferent neuron. This is a different sodium channel. Sour is the most complicated. Let's focus on sour taste cells and not focus on the non-sour taste cells. Let's focus on the sour taste cells. Sourness, acids, are usually weak acids. You know, vinegar, which is a pretty strong acid, is a tastant. We can taste the taste of vinegar. A lot of the other acids that we taste, vinegar is acetic acid. It's a relative, by acid standards, it's a relatively weak acid. And we have vinegar is a dilution of acetic acid. So the acid, as you've learned in chemistry, is, there's the, I've forgotten the language here, there's the acid and the conjugate base of the weak acid. Thank you very much. What do we call this thing that's the pair? The weak acid is neutral, typically. And so it can diffuse right through lipid membranes, where it does its dissociation into the acid and the conjugate base. So the point is the weak acids can diffuse through membranes. They dissociate the acid, the protons, released intracellularly by the influx of this weak acid, block a potassium channel. What does block of potassium channel do to the membrane potential? Depolarizes it. We're blocking potassium efflux, which is a depolarization. Depolarization leading to more neurotransmitter release, leading to more action potentials in the afferent neuron, the secondary cell.
Can you repeat what blocks the sodium channel?
The acid
Okay.
blocks the potassium channel, causing a depolarization. Depolarization, voltage-gated calcium channels, calcium influx, synaptotagmin, snares, neurotransmitter release. Hopefully that's, I'm saying it very fast because that story should be way into your review mode. In addition, acids can go through acid channels themselves. So there are proton channels. So in addition to the acid getting through the lipid, the acid can go through, the protons can go through proton channels where they block the potassium channel and do the same thing. So there's two ways the acid can get into the cell to cause block of potassium channels. As a weak acid through the membrane, and as a strong acid through this acid, proton channel. Let's not worry about what non-sour taste cells do. It's just that they're not geared up for sensing taste, so they have a different response to acid. It turns out that their K channels are not as acid sensitive as the K channels that are expressed in the acid sensing receptor cells. It's pretty simple. So this is the molecular mechanism for transducing tastes into receptors, into depolarizations, release of neurotransmitter, and then into frequency of action potentials from the afferent neuron going either in the glossopharyngeal neuron, the vagus axon, or the facial nerve axon. Going to the brain, going to the, I'm going to go back a few slides, going to the taste cortex in the lateral frontal lobe. Here's the, what's the name of this gyrus right here? I mean the sulcus, what's the name of that sulcus? Central sulcus. So we're in the frontal lobe here, lateral, near the olfactory cortex, but not the same as the olfactory cortex. Okay, so we've converted taste into signals. This is just really a review slide. So this is looking at something that caused depolarization. So the receptor eventually caused depolarization. Depolarization triggered calcium channels that caused synaptotagmin, snares, etc., neurotransmitter release. So this slide just lumps all of the receptors into something that causes depolarization. In this slide, and I'm not sure this is now universally accepted, when they drew up this slide, they thought that the neurotransmitter of taste cells was serotonin. I think there are multiple neurotransmitters that are released by taste cells. But they all, so I'm not going to ask you to memorize this because I'm not sure it's still true, but they all activate the depolarization and action potential frequency in the secondary neuron that takes the action potentials to the taste cortex. If there's no lateral inhibition in the taste pathway. At least not until we get into the brain, where there could be, there are inhibitory interneurons all over the place. And so there could be tuning of taste modalities in the brain, but there's no tuning of taste modalities in the taste bud. Aiden?
Sorry, back in the previous slide, what did you say was happening for the non-sour taste cells again?
The non-sour taste cells have a potassium channel that is relatively insensitive to intracellular acid.
And what is it K block is showing?
Well, that's just showing that there's a little bit of block compared to a lot of block.
And what like below that?
This is an experiment that showed that even in non-sour taste cells, if they used a drug that blocked potassium channels, they were able to turn a non-sour taste cell into a sour taste cell because of the drug. So that's what this is showing. But that's not a key thing. All right, we've got 10 minutes. I know we're all itching for break, but let's go through some of these. We won't go through them all, but there's plenty of them. So we've got some time, so let's do some of the clickers. And some of these might be repeats from some of the clickers from previous classes. Which of the following sensory systems uses GPCRs for signal transduction? Visual system, remember the photoreceptor molecule is a GPCR, but in the auditory system, the hair cell, that's not a GPCR, that's a stretch-gated channel. In the olfactory system, we just talked about GPCRs, but in the somatosensory system, we don't have GPCRs that are transducing touch or pressure or pain. So, room for learning. The GTP-bound alpha subunit of transducin does what? Which sensory modality are we thinking about when we think about transducin? Visual system. In the visual system, the activation of the G protein, transducin, does what? Activates the phosphodiesterase, cuts the cyclic GMP. Well, maybe I'm going to give away the answer to the next. Oh, okay. GTP bound. So there's a story that happened after the activation of phosphodiesterase. Let's just do a few more. I don't want to torture you with these, but let's just do them together. The GTP-bound alpha subunit of G-OLF activates adenylate cyclase. So that was in your brains because we just talked about it. In photoreceptor cells, cyclic G. In photoreceptor cells, cyclic G, which is cut by the phosphodiesterase, does what? Cyclic G activates cation channels, which is why in the dark, the photoreceptor cells are relatively depolarized. The cutting of cyclic G lowers cyclic G, lowers the activation of the cation channel, allowing the hyperpolarization. Maybe this will be the last one. In auditory hair cells, stretch of the tip link I saw a lot of C's. Activates the cation channel, that's what depolarizes the hair cell, causing it to relieve calcium influx, neurotransmitter release. In olfactory cells, cyclic AMP activates cation channels. We just did that. So there's a whole bunch of them. I mean, I'll just show you how many there are. I'm not going to show you. There's scores of these clickers that you can use to review for the next test. But the next test isn't coming up until after break, so it's there for you to work with after break. The next quiz is going to be due the Sunday after break. So you come back to school, we've got a week of break, and then the next quiz, which will focus on anatomy that we covered in class and in the coloring book, is due the Sunday after, not the Sunday of, but the Sunday after.
Does that mean the anatomy of all of the senses?
All of this. Yeah. That Sunday. That's the first Sunday after you get back to school. But not the Sunday before you get back to school. That's great. It's still great.